Hello everyone, my name is Ling Luo. I'm postdoc fellow in Bio-NLP group at NCBI. Thanks for inviting us to present our work in the DRAG project. The DRAG project aims to extract drug-protein interaction from PubMed upstream, providing the gold standard entities. There are a total of 13 relation tabs. According to the future of the corpus, only less than 1% of the relations are crossing multiple sentences in the training set. Therefore, we focus on the training of the relation extraction within the sentence. We developed two independent deep learning-based approaches based on the biomedical pre-trained language models. As shown in the figure, one is the traditional text classification framework. We also explore a novel sequence neighboring framework. Then routing ensemble methods are further used to improve the final performance. In the text classification framework, we treat the text as a multi-class classification problem. We represent a sentence with a drug protein pair using two different ways. The first representation is to insert entity tags in front of the drug and protein mentions. We also treat the entity name tags as the first sentence and use the SEP tag to concatenate it with the first way sequence as the second representation. Then we use biomedical pre-trained model to classify the text into the relation tab. We also propose a novel sequence neighboring framework to address the text different from the text classification. The task is converted to a sequence neighboring problem. Given a candidate source entity in a sentence, the goal of the model is to recognize all the corresponding target entities that are enrolled in the relations. We designed a simple tagging scheme for this task. For the input sequence, we insert argue1 tag pairs in the beginning and the end of the source entity. Besides, drug and pro tag pairs are inserted in the beginning and the end of the drug protein mentions to avoid the entity types and the boundary. For the output sequence, the tokens enrolled in the relation will be tagged by the relation tab, and we use the label O to represent other tokens which do not enroll in a relation. We evaluate the 5 biomedical pre-trained model to the sequence neighbor method. To further improve the performance, we use 3 ensemble methods. For the majority voting, we select the relations that are predicted by more than half of our model. In addition, we search backward and random to find a subset of our model that may achieve higher performance on the development set than using our model. During the task, we submit 5 rounds as our final submission. For round 1, we merge the official training and the development set, then randomly select 350 upstream as our development set for early stopping. Only sequence neighboring models are assembled with the majority voting. In round 2, we use the same model and ensemble method. Difference is we use only the training set for model training and early stopping by the performance on the official development set. In round 3, both the text classification and the sequence neighboring models are assembled by voting with backward search. Round 4, the models are assembled by voting with random search. In round 5, all models above mentioned are assembled by majority voting. Finally, round 5 achieved the highest overall F1 score on the main test site. We also participate in the additional large-scale task. We select 4 eventual models according to their performance on the development set, then use different combinations of them with the majority voting to generate our submission. Each model took about 5 days for operating the large-scale test site on 1 GPU. Similar results are observed on the large-scale test site. In summary, we propose a novel sequence neighboring framework. His performance as good as traditional text classification, even achieved better performance. We tested many biomedical pre-trained language models, and there is no clear winner. The ensemble often can achieve better performance. Now our method doesn't use any additional resource and post-processing. After the release of the gold standard test set, we will do error analysis and we will do more comparison between the two frameworks and try some additional features. Finally, thank the organizer of the task and thanks our team members, my colleague Poting, Zhixuan, and our supervisor Dr. Lu. The research was supported by the NLM. Thank you for your attention. If any questions, welcome to email to me.